dissertation work of mine. So speciation events are depicted as bifurcating processes, and species delimitation has been a historically difficult problem. One of the main reasons for this is because using different criteria or data sources may give us different results. So if we analyze some behavior between two seemingly divergent lineages, we may find that they are truly divergent in terms of their behavior before the speciation event has actually occurred. And alternatively, we may see that morphological characters really only diverge after this divergence event. And so as a partial solution to those difficulties with traditional taxonomy and traditional methods of species delimitation, methods that rely more on gene trees within the species tree or on genetics have become more popular. Specifically, methods that are relying on the multi-species coalescent. And this is what I'm going to talk about today. So often in the literature recently, we're seeing an influx of studies that are looking at purely genetics, inputting those genetic data into these multi-species coalescent-based programs, and changing taxonomy based on those results alone. So typical workflow for these MSC-based methods looks something like this, where we first start off, of course, with samples across the geographical area. Then we gather data from those samples. Usually it's exclusively genetic data, although it doesn't have to be. We use some kind of clustering algorithm to designate some of the population structure within those lineages, or purported lineages. We generate a guide tree using those clusters as terminal taxa, and that guide tree literally guides the MSC-based method. We don't have to use a guide tree, but the example I'm going to use today does use one, so I've included it in this pipeline. And it's from those MSC-based method output that we make species designations. So instead of focusing on these methods themselves and how they work, I think we're going to hear a lot about that later today. I'm going to focus more so on the upstream consequences of changing data and seeing how these methods are changing with a specific case study. With the general question of how sensitive are these MSC-based methods to changes upstream in this workflow? The taxonomic group to focus on today is the American milksnake, Lampropeltis triangulum complex. And these snakes are very widespread, so they range from southern Ontario all the way down into Ecuador. And as you can see, they're very polymorphic. So the ones in southern Ontario are pretty cryptic and brown, but ones that occur sympatrically with coral snakes are very good examples of Batesian mimics. And historically, the taxonomy of this group has changed quite a lot. So some authors, like Williams, thought that triangulum was just one widespread polytypic species with different races represented as subspecies. Many other authors recognize the distinction of Lapsoides, or the scarlet king snake. You can see there's a wide area of symmetry here where the two don't interbreed. Other authors, like Blanchard, have proposed that the complex consists of 3 species, including Polyzona, here in Central South America. And it's important to note that based on our analyses of the existing literature, in terms of morphology, life history, and behavior, this seems to be the hypothesis that goes along with those data. And so, in 2014, the most recent work on the taxonomic group, Ruan et al. Recognized, using genetic data and an MSC-based method, BBP, they recognized 7 species within the complex. So they elevated existing subspecies to full species status. And it's this case that I want to talk about today, using these data from the study. The general question, again, of how robust are these specific results to upstream changes in that MSC-based method pipeline. So Ruan et al., for their workflow, I'm just going to show you a bit of what they did. So they included milksnakes and also other members within the genus, as well as a couple outgroups. For their data, it was mostly nuclear genes with one mitochondrial gene. Their population structure program was Structurama. I'll get into that in 1 second. They generated guide trees and used BBP, as I mentioned, as their MSC-based method. It was mostly from BBP that they made their species-level designations, but not primarily. So firstly, let's look at Structurama. So Ruan et al. This is just a schematic of what they found. They received the highest support for a K, or a number of clusters, of 7. And you can see here that these aren't the 7 species they elevated, because it includes a reference taxon, the gray-banded king snake, or L. alterna. What they noticed is that gentilis and triangulum were sharing a single cluster. So what we decided to do was reproduce these results using all the same data and the same parameters and everything. And when we included all the samples, including those from other members of the genus and the outgroups, our results looked like this. So I'm just going to remove the specimens that don't belong to the complex so we can focus on them today. And what we see is that, just schematically here, Polyzona, Micropholus, and Abnorma are sharing a cluster, and as are Annulata, Gentilis, and Triangulum. We ran this 10 times, and it was converging on basically identical results every single run. And so, of course, these are not the same results as we wanted Al got, so we really wanted to try to reproduce these and figure out how we could reproduce them. One way we were able to do this is if we just considered individuals from within the complex. So we only ran those individuals in the structural analyses. When we did this, you can see we got a K of 7 with the corresponding clusters that Rowan et al. Found. But importantly, this was a single run out of 10, and I could never reproduce it after 9 more runs. I was getting very differing results. So you can see here with K values ranging from 4 to 7, which means that the program wasn't converging, even if I ran it for a very long time. So 2 things to note from here that are consistent are, again, that Polyzona, Micropholus, and Abnorma are forming a single cluster, seemingly, and the Gentilis and Triangulum are as well, like Rowan et al. Found. So this really goes to show that some of these population structure methods, like Structurama, are of course dependent on the samples that you're inputting. And we were getting pretty different results, especially in this case where the results weren't converging, depending on the samples that we were inputting. So the next portion of the workflow involved the use of a guide tree and BPP. And just for simplicity's sake today, I'm only going to present 2 guide trees that I ran. One, Rowan et al.'s results, and the other one, our Structurama results. So if we look at the guide trees, they look like this. And Rowan et al. Received 100% posterior probability score for all of the nodes on their tree. And when we did this, we also got the same. So 100% support for each of these divergences. However, when we run our guide tree, of course, we see 100% for those nodes as well. Meaning that if you're not tweaking these things and you're not testing a million guide trees, you don't really know what's going on with the program. And if you just were to input a single guide tree from your best supported structural result, you would get support for the Blanchard hypothesis of 3 species. So lastly, I mentioned earlier that Rowan et al. Didn't just rely on BPP's results to delimit species. They also wanted to test for gene flow. This is because a lot of these MSC-based methods assume that there's little or no gene flow between purported lineages. And in fact, research has kind of shown that BPP is pretty robust as long as there aren't more than 1 migrant per generation. So Rowan et al. Tested for gene flow using the Migrate-N software, and they concluded that all their comparisons were fewer than 1 migrant per generation, and that BPP's assumptions were not violated. But one important thing that we noticed here was the evenness of the samples, and specifically the distance between closest samples between purported lineages. So if you take a look down here, the distance between Abnorma and Micropholus closest samples is around 36 kilometers. But the distance between closest samples of Gentilis and Annulata is about 530. So of course, when we get migration rates, they're not very comparable if our samples are taken from such different distances. Luckily, Migrate-N can account for geographic sampling distance now. It has the functionality. And so we again reran all the analyses, the same parameters, first without correction for geographic distance. We see our results echo pretty similarly Rowan et al.'s. Ours are shown here in darker gray. And I put a line across for that threshold, that kind of rule of thumb for BPP's assumptions at 1. When we account for geographic sampling distance, we see that the means of all but 2 of our points exceed that threshold. And just as a general trend, we see that kind of the right-hand side of this figure, which represents the comparisons within the US and northern Mexico, are really well above it. So it's likely, based on these results, that the assumptions built within BPP of little or no gene flow between lineages were violated, and that BPP's results are likely not too reliable. So overall conclusions about milksnakes. So first of all the available data really do support only 3 species, and I wasn't able to present the other, morphology, life history, and behavior, but I can talk to whoever after. So we really do find support for triangulum, lapsoides, and polyzona, at least given the existing data. Without further sampling, especially at the contact zones between the purported lineages, it's hard to tell. In terms of general conclusions, I hope today that I've kind of shown you that sampling, population structure inference, and your guide tree input really do strongly affect, in certain cases, the MSC-based method. And that these methods are very useful for forming hypotheses about species limits, but potentially not making taxonomic changes. And so we kind of want to amend this protocol and say that maybe at the end of these MSC-based methods, if you have limited sampling, you can just use this as a hypothesis formation tool. Secondly, we urge caution to researchers, if they're using these MSC-based methods alone, that they ensure that upstream parameters are tweaked to test for the robustness of those results, because clearly it does very heavily impact what you're finding in certain cases. Thank you so much for coming, and I want to thank everyone. Specifically, I want to thank Sarah Rowan, who was very helpful and supportive during this whole thing to help me out with these analyses. I'm happy to take questions.
If you would have run each of the clusters individually in structure alma, would it show structure within each of those groups? Rather than throwing all of the groups in together?
I don't know. I didn't do that, but I can potentially. Yeah, it might just like keep subdividing it. I would kind of assume not though, because when we ran the complex, it didn't even converge on those data. So I just don't think there's enough information in those data. So if there isn't enough information in those data, then further subdividing the sampling, it probably just wouldn't converge. That's a good question. Yeah.
I had a tree that wasn't a species tree, so you just had all your individuals in it. Do all of the 7 or whatever species from the original study each form a monophyletic group? Or are they all mixed up with that original data set?
They don't form a monophyletic group because milksnakes aren't monophyletic, but within the actual purported lineages, no. They don't stick together unless you designate those, in beast, you say, these are my lineages.
Sorry, yeah.
Exactly, yeah. So no, they jump around a lot, especially gentilis and triangulum.
It's interesting that the editors accepted those species designations without strong evidence.
Well, I think that given the analyses that the authors conducted, there was pretty strong evidence. I think if you don't recognize that you need to be tweaking things and checking for robustness, then it does look like the results are pretty good, So it's just, yeah. It's always a case of like trying to figure this stuff out and get these methods working. Okay, thank you.